breakthrough Alzheimer's prevention, clinical trials, and future strategies. Are you ready to revolutionize Alzheimer's research? Discover how cutting-edge clinical trials are shifting from treatment to prevention, offering new hope for researchers and clinicians alike. Recent advancements in Alzheimer's disease research mark a significant transition from managing symptoms to proactively preventing the disease. This shift offers a beacon of hope for millions globally, focusing on halting the progression before irreversible damage occurs. Scientists are exploring innovative early intervention strategies. These include identifying biomarkers years before symptom onset and employing immunotherapies to target the disease's root mechanisms. Understanding these approaches is crucial for researchers and clinicians aiming to combat Alzheimer's effectively. This video delves into groundbreaking prevention strategies, from biomarker-driven detection to vaccine-based therapies. We will examine how these methods overcome traditional treatment barriers and address the challenges of translating lab success into effective clinical interventions. Early detection of Alzheimer's biomarkers provides a critical window for intervention, preventing irreversible neurological damage. Advances in detection technology offer multiple pathways for identifying at-risk individuals during preclinical stages, significantly enhancing prevention efforts. Positron emission tomography, PET imaging, has evolved into a sophisticated tool for tracking amyloid accumulation, a key indicator of Alzheimer's pathology. The amyloid clock concept allows clinicians to measure amyloid plaque burden and predict disease progression with unprecedented accuracy. PET amyloid imaging visualizes protein deposits 15 to 20 years before cognitive symptoms appear. Modern amyloid PET tracers like florbetapir and flutometamol bind specifically to beta amyloid, distinguishing between normal aging and pathological processes. Standardization of centiloid units for quantifying amyloid burden across PET tracers enhances research consistency. This universal scale facilitates direct comparisons between studies, accelerating the validation of prevention protocols. However, PET imaging's clinical application is limited by cost, radiation exposure, and availability. Blood-based biomarkers represent a major advancement in accessible Alzheimer's screening. Phosphorylated tau at position 181, p-tau 181, demonstrates remarkable specificity for Alzheimer's pathology, increasing in blood plasma years before symptom onset. Glial fibrillary acidic protein, GFAP, indicates astrocytic activation during early disease stages. Rising GFAP levels reflect the brain's inflammatory response to amyloid accumulation, often preceding tau pathology. Combining p-tau 181 and GFAP measurements provides a comprehensive view of pathological processes. Multiplex assays can simultaneously detect multiple blood biomarkers, improving diagnostic accuracy. Ongoing standardization efforts promise to make these tests universally available for clinical practice, enhancing early detection capabilities. Measuring formic acid in urine samples offers a completely non-invasive screening method. This metabolic byproduct increases significantly during early Alzheimer's pathology, providing opportunities for widespread screening. Urine formic acid testing offers several advantages, including easy sample collection and promising sensitivity in distinguishing between healthy controls and individuals with mild cognitive impairment. Longitudinal monitoring can be conducted affordably, allowing for regular risk reassessment. Mass spectrometry techniques have refined formic acid detection methods, enabling precise quantification from standard urine samples. Portable testing devices under development may soon allow for point-of-care screening in primary care settings, revolutionizing early detection. Integrating these biomarker approaches creates a comprehensive framework for early Alzheimer's detection. Clinical trials can now enroll participants at precisely defined biological stages, fundamentally changing prevention study design and improving outcomes. Designing effective clinical trials for Alzheimer's prevention requires methodical approaches that differ notably from traditional treatment studies. The shift toward prevention necessitates specialized trial structures, carefully defined inclusion criteria, and strategic risk stratification methods to identify those most likely to benefit from early interventions. Prevention-focused Alzheimer's trials follow standard pharmaceutical development phases but with important adaptations. Phase 1 trials typically involve small volunteer groups to assess safety and appropriate dosage. Phase 2 trials expand to hundreds of participants, evaluating both effectiveness and continued safety monitoring. Finally, Phase 3 trials scale to thousands of participants to confirm efficacy across diverse populations. The AHEAD 345 study exemplifies innovative trial design through its two-trial platform approach. This 4-year study tailors anti-amyloid antibody dosing regimens based on baseline PET amyloid levels. The A45 arm, Phase 3, includes individuals with elevated amyloid, greater than 40 centiloids, using the preclinical Alzheimer's cognitive composite, PACC5, as the primary outcome, whereas the A3 arm, Phase 2, includes those with intermediate amyloid 20 to 40 centiloids, with amyloid PET change as the primary outcome. Other groundbreaking prevention trials include studies targeting individuals with dominant genetic mutations. For instance, the Dominantly Inherited Alzheimer Network, DIAN, enrolls patients with presenilin 1, presenilin 2, or amyloid precursor protein mutations. In due time, the Takeda Zinfandel study will follow 6,000 participants for 5 years, using age, ApoE4 carrier status, and TOM40 gene status to stratify risk groups. Establishing precise inclusion criteria is vital for early intervention trials. For mild cognitive impairment MCI studies, participants typically show objective impairment on psychometric tests without affected daily activities. Common requirements include scores of 24 to 30 on the Mini Mental State Examination (MMSE), 0.5 on the Clinical Dementia Rating Scale (CDR), and memory box scores greater than or equal to 0.5. Biomarker criteria have become increasingly important for identifying appropriate candidates. At first, trials primarily used amyloid PET or cerebrospinal fluid (CSF) markers as inclusion criteria. In spite of their value, stringent biomarker criteria contribute to high screen failure rates, 88% in preclinical and 78% in prodromal AD trials. Moreover, most trials require study partners who can report on participants' daily cognition, which further limits eligible populations as many potential participants live alone. ApoE genotyping has emerged as a crucial tool for risk stratification in prevention trials. As the strongest genetic risk factor for late-onset Alzheimer's, ApoE status notably influences trial design and participant selection. ApoE epsilon 4 carriers face substantially higher risk, with homozygotes experiencing the greatest risk elevation. Trial randomizations are regularly stratified by ApoE epsilon 4 status, carriers versus non-carriers, and geographical region. This stratification is especially important given that ApoE epsilon 4 carriers show higher rates of amyloid-related imaging abnormalities (ARIA). For example, in the Lecanemab trial, ARIA-E occurred in 32.6% of homozygotes compared to just 5.4% in non-carriers. Advanced approaches now combine ApoE with other genetic variants in polygenic risk scores, PRS, to identify high-risk individuals. These scores can predict earlier onset with up to 9 years difference in median age between highest and lowest risk groups. Immunotherapy has emerged as a frontline approach in Alzheimer's disease research, with remarkable progress in targeting both amyloid and tau pathologies. Recent clinical trials demonstrate that certain antibodies can not only clear pathological proteins, but also slow cognitive decline when administered before extensive neurodegeneration occurs. Monoclonal antibodies targeting amyloid beta have shown promising results in recent Phase 3 trials. Lecanemab, tested in the Clarity AD trial, demonstrated a 27% slowing of clinical decline over 18 months compared to placebo. After 18 months of treatment, participants' mean amyloid levels dropped to 22.99 centiloids, below the 30 centiloid threshold considered indicative of elevated brain amyloid. Likewise, Donanemab showed marked efficacy in the Trailblazer ALZ-2 trial involving 1,736 participants with early Alzheimer's. The least squares mean change in integrated Alzheimer disease rating scale score was -6.02 for donanemab versus -9.27 for placebo in the low-medium tau population, representing a 35% slowing of decline. Both treatments show greater efficacy in earlier disease stages. Indeed, donanemab slowed decline in memory and thinking by approximately 35% compared to placebo, specifically in people with low-medium tau levels. Moreover, almost half, 47%, of those receiving donanemab showed no major symptom worsening over 1 year, compared to 29% on placebo. Notwithstanding these benefits, safety concerns exist. Amyloid-related imaging abnormalities, ARIA, occurred in 32.6% of lecanemab-treated participants. Similarly, brain swelling appeared in approximately 25% of donanemab recipients, with 5% experiencing symptoms. ACI 35.030 represents a groundbreaking active immunization approach targeting phosphorylated tau, p-tau. In recent trials, this liposomal vaccine generated antibody responses in 100% of older patients with early Alzheimer's disease. Subsequently, anti-p-tau IgG response was observed after the first injection, with very high titers achieved following injection. Essentially, ACI 35.030 contains a liposomal formulation incorporating a conformationally constrained p-tau peptide antigen, adjuvants, and non-tau T helper peptides. The antibodies generated preferentially target phosphorylated tau over non-phosphorylated forms with sustained response for more than a year. Safety data has been encouraging, with no clinically relevant safety concerns detected. Accordingly, researchers have initiated a Phase 2b trial called RETAIN, designed to test the hypothesis that ACI 35.030 has disease-modifying effects that can delay or prevent cognitive impairment. A novel approach to tau clearance involves nasal delivery of toxic tau conformation-specific monoclonal antibody 2, TTCM2. This antibody selectively recognizes pathological tau aggregates in brain tissues from patients with Alzheimer's disease, dementia with Lewy bodies, and progressive supranuclear palsy. Researchers found that intranasally administered TTCM2 loaded in mice cells, TTCM2-MS, efficiently entered the brain in mouse models, targeting pathological tau in intracellular compartments. Remarkably, a single intranasal dose effectively cleared pathological tau, elevated synaptic proteins, and improved cognitive functions in aged tauopathy mice. The mechanism involves tripartite motif containing 21, TRIM21, an intracellular antibody receptor that facilitates proteasomal degradation of cytosolic antibody-bound proteins. TRIM21 proved essential for TTCM2 MS-mediated clearance of tau pathology, highlighting a novel pathway for targeting intracellular tau aggregates. Beyond immunotherapy approaches, cutting-edge Alzheimer's disease research has identified specific molecular pathways that offer favorable prevention targets. These novel interventions focus on addressing the disease's cellular mechanisms before extensive brain damage occurs. Mitochondrial dysfunction represents a critical trigger in Alzheimer's pathogenesis. The voltage-dependent anion channel 1, VDAC1, protein serves as a mitochondrial gatekeeper controlling cellular metabolism and calcium homeostasis. In Alzheimer's, VDAC1 becomes overexpressed in brain tissue surrounding amyloid plaques, leading to neuronal death. A novel compound called VBIT4 prevents VDAC1 oligomerization and blocks its pro-apoptotic activity. In neuronal cultures, VBIT4 successfully prevented amyloid beta-induced cell death. Most impressively, oral administration of VBIT4, 20 mg per kilogram twice weekly, rescued cognitive function in 5 times FAD mice as measured through 4 behavioral tests. This treatment additionally protected against synaptic and neuronal loss in both cortex and hippocampus regions. Histone acetylation imbalances represent another promising intervention target. The TYP60 histone acetyltransferase enzyme, typically depleted in Alzheimer's brains, plays a dual role in both genetic expression and RNA processing. Researchers discovered that TYP60 binds to RNA in the brain, controlling how RNA molecules are spliced to generate diverse proteins. This function proves particularly important since splicing errors contribute markedly to Alzheimer's pathology. Restoring TYP60 levels in Alzheimer's models not only rescues gene activation but also partially protects against splicing disruptions. A groundbreaking targeted protein degradation approach focuses on phosphorylated P38 MAPK PP38, a modified protein involved in Alzheimer's inflammatory processes. The compound PRZ1800 selectively binds to and degrades PP38 while sparing normal P38, making it highly specific even when tested against 96 different protein kinases. In 5x FAD mice, intranasal PRZ18002 treatment remarkably reduced brain PP38 levels, decreased neuroinflammation, and lowered amyloid beta deposition. These biochemical improvements translated to functional benefits, with treated mice showing enhanced spatial learning and memory performance. This innovative approach highlights the potential of targeting specific post-translational modifications in Alzheimer's prevention. Despite favorable laboratory results, Alzheimer's disease research faces substantial challenges when translating preclinical successes to effective human treatments. The staggering failure rate of clinical trials, approximately 99.6%, highlights the significant disconnect between animal models and human pathology. Current transgenic mouse models mimic certain pathological features of Alzheimer's disease but fail to capture its full complexity. Although these models develop amyloid deposits, they typically lack widespread neurodegeneration and regional brain atrophy that characterize human Alzheimer's. In reality, animal models show only minor localized neurodegeneration, even in very old mice. Perhaps the most notable limitation involves neurofibrillary tangles, NFTs. Though some mouse models exhibit localized hyperphosphorylated tau representing pretangles, none naturally develop true NFTs without incorporating mutations not associated with human Alzheimer's disease. Many widely used tau transgenic models rely on P301L or P301S MAPT mutations, which cause frontotemporal dementia rather than Alzheimer's. Due to these shortcomings, animal models should be considered models of AD-like pathologies rather than comprehensive disease models. Even aged non-human primates developing modest amyloid deposition show limited evidence of additional Alzheimer's pathologies. The blood-brain barrier, BBB, represents another formidable obstacle, preventing over 98% of potential drugs from reaching brain tissue. This natural filtration system particularly restricts antibody penetration, allowing only a small fraction of intravenously administered treatments to enter the brain. Consequently, achieving optimal concentrations often requires higher doses and more frequent treatments, creating potential toxicity and side effects throughout the body. In the case of monoclonal antibody treatments, these higher doses can trigger amyloid-related imaging abnormalities, ARIA, causing brain swelling and potential bleeding. This challenge has spurred exploration of alternative delivery approaches. These include non-invasive techniques like exosome-based delivery systems, small enough to pass through the blood-brain barrier, and focused ultrasound technology that temporarily opens the blood-brain barrier when combined with microbubble injections. Unfortunately, even when drugs successfully cross the blood-brain barrier, they face additional hurdles, including P-glycoprotein efflux pumps that actively transport foreign substances out of the brain. Understanding these translational limitations will help researchers develop more effective prevention strategies that account for both the biological complexity of Alzheimer's disease and the physical barriers to drug delivery. Alzheimer's disease research has undergone a fundamental paradigm shift, moving from reactive treatment approaches to proactive prevention strategies. Early biomarker detection creates unprecedented opportunities for intervention decades before cognitive symptoms appear. Immunotherapy approaches have demonstrated great potential in clinical trials. Lecanemab and Donanemab have shown the ability to slow cognitive decline by targeting amyloid deposits, while innovative tau vaccines like ACI 35030 and nasal TTCM2 antibodies offer complementary strategies. Targeting specific molecular pathways presents another promising frontier. VBIT4's protection of mitochondrial function, TIP60 restoration to prevent RNA splicing errors, and PRZ1802's selective degradation of inflammatory proteins each offer distinct mechanisms for disease modification. Monumental challenges persist in translating these breakthroughs to widely available human treatments. The limitations of animal models and the formidable blood-brain barrier continue to complicate drug development and delivery. Despite these obstacles, the convergence of multiple prevention strategies suggests we are closer than ever to meaningful interventions. As research advances, the integration of these complementary approaches may finally provide effective prevention options for the millions at risk of developing Alzheimer's disease. The transition from treating symptoms to preventing their occurrence represents not just scientific progress, but renewed hope for patients, families, and healthcare systems worldwide. Ready to take the next step in Alzheimer's research? Subscribe to our channel for more in-depth analyses, cutting-edge research updates, and expert insights. Share this video with your colleagues, and let's work together to revolutionize Alzheimer's prevention. Click the link in the description to access the complete blog article.